Have you ever stopped to wonder why we seek the company of others? From a bustling city square to a quiet family gathering, humans are drawn together. We are, by our very nature, social creatures. We find comfort, safety, and joy in being part of a group. This deep need to connect is not something unique to us. In the meadows and gardens, another remarkable society thrives. The honeybee, a tiny insect with a mighty community, also builds its life around cooperation and togetherness. They work as one, communicate constantly, and depend on each other for survival. We see this instinct to gather in so many corners of the natural world, a shared thread that connects vastly different forms of life. Yet, even within the most tight-knit groups, differences appear. Not every person is a social butterfly, and not every bee is the busiest worker in the hive. Some of us are naturally more outgoing, while others prefer quiet solitude. This variation in social behavior is completely normal. Sometimes it is simply a matter of mood. We might feel more sociable on a sunny day than on a gloomy one. Our past experiences also shape how we interact with others, teaching us who to trust and how to form bonds. But scientists are now discovering that something much deeper, something written into our very code of life, also plays a crucial role. Our genes can influence just how much we desire to be with others. This idea that our social tendencies are partly inherited is fascinating. It suggests that our personalities are not just formed by our choices or our environment alone. An ancient, invisible force guides our actions. The same is true for the honeybee. Within a single hive, some bees are more interactive than others. Some eagerly share food and information, while their sisters might be more reserved. For a long time, we could only observe these differences. But now, thanks to wonderful new technology, we can begin to understand the reasons behind them. We are learning that the desire to form friendships is not just a feeling, it is a biological instruction. The parallels between a human city and a beehive are extraordinary. Both are complex societies where individuals play different roles, yet all contribute to the well-being of the whole. In our world, we have leaders, helpers, and innovators. In the hive, there are foragers, nurses, guards, and of course, the queen. The intricate dance of cooperation allows both humans and bees to achieve incredible things that no single individual could accomplish alone. By studying the simple yet profound social life of the honeybee, we can gain a clearer window into the very origins of our own need for community. It is a journey that takes us deep inside the hive and deep inside ourselves. To understand the hidden rules of bee society, scientists at the University of Illinois embarked on a remarkable study. They needed a way to watch thousands of bees and know exactly who was interacting with whom. How could they possibly keep track of so many individuals? The solution was brilliantly simple. They attached tiny, lightweight barcodes to the backs of hundreds of honeybees. Each tag was like a miniature name badge, unique to that one bee. This allowed automated cameras and computers to follow every single bee as it moved through the hive, creating a detailed map of their social lives. The scientists could now see who was popular, who was a loner, and how friendships formed over time. With their bees all tagged and accounted for, the researchers focused on one specific behavior. This behavior is a key indicator of friendship in the bee world. It is called trophallaxis. This is the act of sharing food directly from mouth to mouth. When one bee returns to the hive with a full belly of nectar, it will often share its meal with a hungry sister. This is more than just a simple transfer of nutrients. It is a fundamental social interaction. It is how bees strengthen their bonds, pass information through the colony, and ensure that the entire community is fed. By tracking every instance of trophallaxis, the scientists could measure how socially connected each bee was. The automated system worked tirelessly, day and night, recording thousands upon thousands of these food-sharing moments. The data it collected was incredibly rich. It revealed a complex social network within the hive, with some bees acting as popular hubs, sharing food with many different individuals. Other bees were far more selective, interacting with only a small circle of hive mates. It became clear that, just like in human societies, there was a wide spectrum of social behavior. The barcodes allowed the team to move beyond simply observing the group, and to start understanding the individual personalities that make up the whole community. It was a powerful new way to listen to the secrets of the hive. This meticulous tracking was only the first step of the puzzle. Once the researchers had identified the most and least social bees, they could ask a deeper question: what makes them this way? Was it a result of their age, their role in the hive, or something else entirely? The study was designed to find the answer by looking at the bees' genetic makeup. By linking specific behaviors to the genes that drive them, the scientists hoped to uncover the molecular foundations of social life itself. The tiny barcodes were the key that unlocked the door to a world of genetic discovery, linking a simple act of sharing to the ancient code of life. After carefully observing the bees, the scientists moved to the next phase of their investigation. They analyzed the DNA of the bees, looking for clues that could explain the differences in their social behavior. What they found was truly astonishing. The study identified 18 specific genetic variations that were directly linked to how often a bee engaged in food sharing. 2 genes in particular stood out from the rest. These genes are known as neurolegin 2 and NMDAR2. The bees that were the most generous food sharers, the social connectors of the hive, often had specific versions of these genes. It was clear evidence that a bee's tendency to be friendly was written in its DNA. The discovery became even more profound when the researchers compared these bee genes to genes found in humans. They found that neurolegin 2 and NMDAR2 in bees are incredibly similar to genes that exist in our own bodies. In humans, these corresponding genes are known to play a vital role in how our brains develop and function. Even more specifically, they have been linked to conditions that affect social interaction, such as autism spectrum conditions. This parallel is extraordinary. It suggests that the same fundamental genetic tools are being used to build social behaviors in both a tiny insect and a human being, despite our species being separated by over 600 million years of evolution. Furthermore, the study looked not just at the genes themselves, but at how active they were in the bees' brains. The researchers examined the brains of the most social bees and compared them to the brains of the less social ones. They found that the bees who engaged in more food sharing had heightened activity in their brains. In fact, over 900 different genes showed higher levels of expression. Many of these active genes are responsible for building and maintaining the connections between brain cells. This indicates that being social requires more brain power. The brains of friendly bees were literally working harder to manage their complex social lives. This connection between genes, brain activity, and social behavior paints a beautifully complete picture. It shows us that social interaction is not a vague or random event. It is a biological process, guided by a genetic blueprint and powered by the intricate wiring of the brain. The same molecular machinery that helps a bee decide to share its food with a hive mate may also influence how we form friendships, build communities, and navigate our own social worlds. It is a humbling reminder that at the most fundamental level, the drive to connect is a shared feature of life on Earth, sculpted by the same ancient genetic instructions. The discovery that humans and bees share a genetic basis for sociality points to a truly grand idea in biology. This idea is known as the ancient molecular toolkit. Imagine evolution as a builder. Over hundreds of millions of years, this builder has been faced with similar problems in many different animals. One of those recurring problems is how to organize a society. Instead of inventing a completely new solution every single time, evolution has often reached back into its toolkit and reused the same trusted parts. The genes for social behavior, like neurolegin 2, are some of these ancient reliable tools. They have been used again and again to help create social life in countless species. This genetic toolkit dates back to a common ancestor that lived long before bees or humans ever existed. Over 600 million years ago, a simple organism possessed these genes, which likely performed a basic function related to how cells sense and respond to their environment. As life evolved and became more complex, these same genes were adapted for new purposes. In one branch of the tree of life, they were shaped to help build the social networks of an insect colony. In another, they were modified to help shape the social behaviors of primates, eventually leading to the complex societies we see in humans today. The function has changed, but the core components have remained. This concept helps to explain why we see such surprising similarities across the animal kingdom. It tells us that the feeling of belonging we get from being with friends is not just a human experience. It is an echo of a biological process that is ancient and widespread. The honeybee, with its intricate dances and selfless cooperation, becomes a living window into our own evolutionary past. By studying its genes and behaviors, we are, in a way, studying a version of the blueprint that was used to construct our own social instincts. It shows that nature is wonderfully efficient, repurposing its best inventions for all sorts of different creatures. As we learn more about this shared genetic heritage, we are left to ponder a fascinating question. How much of our modern lives, our friendships, our communities, our very societies, is still being guided by this ancient toolkit? When you choose to spend time with friends or feel the comfort of being part of a group, Are you simply feeling the effects of these age-old genes at work? The bond between two bees sharing a drop of nectar and two humans sharing a conversation may be separated by an immense evolutionary gulf, but it seems they are united by a common genetic thread, woven through the deep history of life itself.